Ever wonder how a generic pill can really be considered the same as like the original brand name version?
Well, it's a great question, and there's a lot of science that goes into ensuring that. And one of the most important tests is called dissolution testing.
Dissolution testing.
It's kind of a mouthful, but it's actually super fascinating.
Okay, so in this deep dive, we're going to be talking about dissolution testing.
That's right.
We'll be taking a behind-the-scenes look at how scientists use this in vitro test to basically predict what happens in vivo, inside a living organism.
Yeah, exactly.
So what sources are we going to be using for this?
So our primary source is actually a transcript from Season 9, Episode 5 of this very podcast. It's called Dissolution Testing and Bioequivalence.
Okay, and is there anything else that we'll be referencing?
Well, we also have some snippets from Season 3 on basic pharmacokinetics and a little bit from Season 5, which focused on pharmaceutical product development and something called in vivo correlation.
All right, so today we're going to be looking at essentially how drugs get absorbed in the body.
Yeah, that's a big part of it. We're going to specifically focus on dissolution testing, different types of equipment, why the liquid or medium used in these tests is so important, and how those results are interpreted.
Okay, and you said that this was vital in regulating the approval of new drugs.
Yeah, it's super important for both new drugs like NDAs, new drug applications, and also generic drugs, which go through what's called an ANDA, an abbreviated new drug application.
Okay, so how about we start with the basics? Can you explain the link between this thing called dissolution and bioavailability?
Sure. So bioavailability essentially refers to how much of a drug actually reaches the bloodstream and how quickly it gets there.
Okay.
And for solid dosage forms, like the tablets or capsules that many of us take, the rate at which that dissolves, that solid form dissolves, really impacts how well it gets absorbed in the GI tract, the gastrointestinal tract.
So does that mean that a pill that dissolves quickly is going to be absorbed more quickly than one that dissolves slowly?
Exactly. And that absorption rate directly affects the drug's bioavailability. It determines how much of the drug actually gets into your system to do its job.
That makes sense. So are we essentially saying that dissolution testing is like a predictor of how well a drug will actually work in the body?
You got it. In fact, in 1998, researchers Dressman and his colleagues actually described dissolution testing as a prognostic tool for oral drug absorption. That phrase really captures its importance.
A prognostic tool, huh? That sounds pretty serious. So are there different ways to carry out these dissolution tests?
There are. A variety of specialized apparatus are used to kind of simulate what happens in our stomachs and intestines.
So it's not just like dropping a pill in a glass of water.
No, not at all. These are carefully controlled environments. Different apparatuses are used depending on the type of dosage form you're testing. Like an immediate relief tablet versus a capsule versus a suspension will all be tested slightly different.
So walk me through this. What are some of the key things that scientists consider when they're setting up a dissolution test? Like what kind of things can affect how a drug dissolves?
Sure. So first, there are the properties of the drug substance itself. Think about its solubility, for example. Some drugs are more soluble in water than others. Then there's the dosage form itself, how the tablet or capsule is made.
Right.
And this includes the inactive ingredients or excipients that are used. These can influence things like how quickly a tablet breaks apart or disintegrates.
Oh, I see.
And then, of course, there's the environment of the gastrointestinal tract.
Right, like the stomach acid and all that.
Exactly. The pH of the stomach is very acidic, while the intestines are more alkaline. And all of these factors together, the drug substance, the dosage form, and the GI environment, play a role in how a drug dissolves.
So it sounds like choosing the right liquid for these dissolution tests, the medium, is super important.
It's crucial. The dissolution medium is carefully selected to mimic the fluids in the GI tract.
Like the stomach acid we talked about.
Exactly. For example, they might use a solution with a very low pH, like around 1 or 2, to simulate the acidic stomach environment.
Wow, that's pretty acidic.
It is. Or they might use buffered solutions with a pH closer to 4.5 or 6.8 to represent different parts of the intestines.
Got it. And how does the pH actually affect the drug dissolving?
Well, the pH can have a huge impact on a drug's solubility. Some drugs dissolve much better in acidic conditions, while others need a more basic or alkaline environment.
So choosing the wrong pH could really mess up the results.
It absolutely could. It wouldn't accurately reflect how the drug would behave in the body. And we actually have an example of this in one of our sources. They talk about how some natural and synthetic gums, which are often used as binders in tablets.
Binders.
Yeah, binders are inactive ingredients that basically help hold the tablet together.
Okay.
So these gums can actually form these kind of viscous solutions in gastric fluid, like a gel almost. And that can slow down the dissolution process.
So even something that seems as simple as the binder in a tablet can influence how the drug is released.
Exactly. It all comes down to carefully selecting the right dissolution medium to really get a good prediction of what's going to happen in the body.
Okay, so let's say they've run the dissolution test, they've got all their data. How do scientists actually interpret those results? What are they looking for?
Well, the results are usually presented as what's called a dissolution profile. It's basically a graph that shows the percentage of the drug that's dissolved over time.
A graph. Okay. So are they looking for a specific shape or pattern in that graph?
Well, they're looking at the rate and the extent of dissolution, how fast and how much of the drug is released from the dosage form.
And how does this relate to the idea of bioequivalence?
Well, when you're comparing, let's say, a generic drug to a brand name drug, one of the key things you want to see is that their dissolution profiles are similar.
So if the graphs for the generic and the brand name drug look pretty much the same, that's a good sign.
Exactly. It suggests that they're both releasing the drug in a similar way, which supports the idea that they'll be bioequivalent, meaning they'll have the same therapeutic effect.
So dissolution testing is like a key piece of evidence that the FDA would look at when they're deciding whether to approve a generic drug.
It's a very important piece of evidence. It helps them ensure that the generic drug is truly comparable to the brand name drug in terms of how it behaves in the body.
Mm.
And there's actually a specific regulatory expectation that lots, different batches of a drug,
Right.
even those that are at the upper and lower limits of what's allowed for dissolution, they should ideally be bioequivalent.
So they're not just testing one batch. They're making sure that any batch of that drug is going to be consistent.
Precisely. It's about ensuring consistent quality and performance across the board. And this whole concept of bioequivalence is really at the heart of the regulatory process for ANDAs, abbreviated new drug applications, which is what generic drugs go through.
Right. So for a generic drug to get approved, it has to prove that it's essentially the same as the brand name drug.
That's the main goal. They have to demonstrate bioequivalence. And dissolution testing is often a major part of that demonstration.
Okay, now what about new drugs? You mentioned NDAs earlier. Does dissolution testing play a role there as well?
It does. For new drugs, dissolution testing is crucial for characterizing the drug product. It helps scientists understand how the drug is released from that specific formulation and how it might behave in the body.
So for a new drug, it's not about comparing it to something else. It's about understanding its own unique properties.
Exactly. It's about building a solid foundation of knowledge about that particular drug product. And this data is important for things like setting quality control specifications during manufacturing.
So they can make sure that every batch of the new drug is consistent.
That's right. And it also helps support the overall efficacy and safety profile of the new medication.
Okay, so we've talked about the principles, dissolution testing. We've talked about how it's used in the regulatory process. Do you have any real-world examples of how this testing has actually made a difference? Like, has it ever led to changes in how a drug is formulated or helped get a generic drug approved?
It's interesting. The sources that we have for this episode don't really delve into specific named case studies, but we can definitely talk about some hypothetical scenarios that are very realistic and reflect how dissolution testing works in the real world.
Okay, I like hypothetical scenarios. Let's hear one.
All right, so imagine a company that's developing a new tablet. They're in the early stages of development. They've got this promising drug compound, but when they do their initial dissolution test, they find a big problem. The drug is just not releasing from the tablet very well.
Oh, no.
Yeah, it's dissolving too slowly in the simulated gastric fluid. And that's a red flag because it suggests that the drug might not be absorbed very well in the stomach when someone actually takes it.
So what did they do?
Well, this is where dissolution testing really guides the development process. They know they need to go back and tweak the formulation. They might experiment with different excipients, different inactive ingredients, maybe try a different disintegrant, which is something that helps the tablet break apart more easily, or they might try a different binder.
So they're using the results of the dissolution testing to improve the actual design of the tablet.
Exactly. It's an iterative process. They'll make changes, retest, make more changes, retest, until they find a formulation that releases the drug at the desired rate.
That's really cool. It shows how this testing isn't just a box to check, it's actually driving innovation.
Absolutely. And now let's think about a hypothetical example for generic drugs. Let's say a company is developing a generic version of a popular medication. They've created their formulation, and now they need to prove to the FDA that their version is bioequivalent to the original.
Okay.
So they're going to conduct a bunch of dissolution tests, comparing their generic drug head-to-head with the brand name drug. They'll use carefully chosen dissolution media, the right apparatuses. Everything's got to be just right to mimic the conditions in the body.
Makes sense.
And if the results show that the generic drug dissolves in a very similar way to the brand name drug, then they have strong evidence to support their claim of bioequivalence.
And that's ultimately what gets them approved.
That's a big part of it. The FDA needs to be confident that the generic drug will essentially work the same way as the brand name drug. And dissolution testing is a key piece of that puzzle.
This has been super interesting. So as we wrap up, can you just give us like the big picture takeaway messages about dissolution testing?
Sure. I think the main thing to remember is that dissolution testing is a bridge. It connects what happens in the lab with what happens in the human body.
A bridge. I like that.
It allows scientists to predict how a drug is going to behave once it's taken. And that's essential for ensuring the quality, consistency, and ultimately the effectiveness of our medications.
And it's important for both brand new drugs and generic drugs.
Absolutely. It plays a vital role in the approval process for both, helping to guarantee that patients have access to safe and effective treatments.
You know, it's amazing to think about all the science and the testing that goes on behind the scenes just to make sure that the pills we take actually work the way they're supposed to.
It's pretty remarkable.
And it makes you wonder what other tests are out there that we don't even know about, all working to ensure the quality and safety of our medicines. It's something to think about. Thanks for joining us for this deep dive into dissolution testing and bioequivalence.
Thanks for having me.
This has been The Deep Dive.
Bye.